Okay, so good afternoon. My name is Paul Debbie. I'm the Director of Tech Transfer here at BTI. Today, I'm going to tell you a story about two new companies that are based on BTI technology that we're in the process of starting up here at the Institute. But there's a couple of things I want to tell you before I jump right into those two case studies. So let me give you just a brief outline of what I'm going to talk about. I'm going to talk about something called the Valley of Death. And so this isn't the biblical Valley of Death, but a different valley of death that you hear about when you talk about business concepts. And then I'm going to just very briefly touch on one of the objectives from our strategic plan here at BTI and where new companies fit into that particular plan. And then I'm going to talk about two newcos. That's the term that entrepreneurs use when they talk about new startup companies. And so moving on to the Valley of Death. In business startups, there's a concept that we call the Valley of Death. And I put up a picture here of I think there's a little tag on it here. Oops, sorry, I'll go back. There's a concept that we call the Valley of Death. And this is a picture of the world's highest suspension bridge. It's a pedestrian suspension bridge. This is in a town in China that I can't pronounce, but I'll show you it in a second. And the concept here is that here at BTI, we do very basic research, like you see here on this side of the valley. And in order to have a successful product that goes to market, you've got to get it across this valley. And what happens here is really, you've got a basic technology that's really not a product that's ready for market yet. And then, like I said, over here you have marketable products. But there's sort of this product development process along the way. And this is where products or technology usually kind of dies as it tries to get from here to here. And so one of the ways let me step back just for a second. There's three ways that we license technology or deal with a technology transfer at BTI. There's a number of different ways, but it basically boils down to three ways. The first one is what I call here traditional out-licensing. So that's where a company comes to an institute like ours, and there's some technology that's fairly well developed that they're very interested in licensing from us. So it's like the perfect case scenario for us, right? They just come in, we work out a royalty rate with them, we let them go, sell the product, develop the product, sell the product, and we just wait to rake in the money, right? That's not very common in a basic research setting because most of the technology is not really that developed. We've been lucky here, as many of you know here at BTI, to have out-licensed an insect cell line that's used for protein production. It was used for Cervarix, the HPV, one of the HPV vaccines that's on the market. And that's a very traditional out-licensing of technology. But again, there you had an insect cell line, which was really the commercial labs could just take it up and use it, with the current technology that they already had for protein production. But most times it doesn't work out like that. So sort of an intermediary between beyond traditional out-licensing are what we call collaborative research and licensing agreements. So that's where we may bring in a commercial partner who wants to fund research here at BTI or research at their own institution. And maybe we're doing work with them to co-develop a product to the point where it might be more marketable and ready to be brought in-house as a project at a company. And that usually involves a licensing agreement and maybe some negotiated royalty rates ahead of time, and usually involves money that may come to BTI for our research, or they're also spending money on those technologies at their own place. And we've had a number of different collaborative research agreements here in the history of BTI. The most recent one that I've talked about is one that we had with BASF that's related to one of the concepts I'm going to tell you about here. For plant protection. In the past, we had a project with Dow AgroSciences to develop plant-made vaccines, and we actually developed the first USDA licensed plant-made vaccine as part of that particular project. So it can be a very successful way to move technology along. But the third way that I want to talk about today is the idea of starting up a company around that project and then marketing that technology in sort of a different way. And I'll explain as I go through the two examples how that works. One of the reasons we're doing this more often is because if you look in biotechnology or in the pharmaceutical industries, for example, it's very common for these companies to not buy or license technology. They buy companies that have technology. And so you move it into a company, you kind of move it a little bit further across this bridge to the point where it becomes more attractive or a little less risky for the company to come in and invest. And then they'll scoop it up when they feel more comfortable. Or it may move into a collaborative research and licensing agreement with the new company. There's sort of a blend of how things can sort of take place. But those new companies do give you a little bit more leverage and a kind of a different way to market technology. So here's the name of the town here in China where this is, in case anybody knows. I don't. Like I said, I want to briefly talk about strategic planning. This is I'm not going to read the whole slide, but one of our later objectives in our strategic plan is obviously focused around translating discoveries to real-world applications and value. This is a very important part of BTI's mission, and tech transfer is an important part of trying to do that, getting our science out into society in a way that can have a positive impact. And like I said, the way we do that is partnering with companies, out licensing or collaborative research agreements as part of that partnership, or startup companies. So we're specifically focused now on trying to do some of these startup projects. And now I'm going to tell you about two of them that we have in the hopper right now. First, I thought this is kind of a fun cartoon here. It's a guy coming in for a job interview at a startup, and they're saying, since we're a startup, we were hoping you would pay us to work here. So startup companies are a very risky area to get into. They can be very rewarding for people that have sort of that risk-averse personality, but it really is a high-risk area. Most of these companies, probably more than 90% of them fail within the first 5 years. The trick is to find the small percentage of the ones that do, and then they can tend to be very successful. But that's kind of the world you're walking into when you start talking about startup companies. Okay, so I'm going to give you two examples. I'm going to talk first about a little bit more developed project that we have, which is a new company developing novel treatments, seed treatments to protect agricultural crops. And then I'm going to talk about a newer company that we're looking into for developing technology to improve recombinant protein production in plants. And don't worry if that doesn't make any sense to you. I'm going to try to explain that as I talk about that particular project. And as I talk about each one, what I'm going to try to do is I'm going to talk about the concept and try to explain to you the technology behind it so that you can understand how it works. Then I'm going to talk and show a little bit of the proof of concept data that we have for each one of these technologies. They're not just a vision. We actually have some work that we've done, and we know that it could be promising. Then I'm going to give a status update on each of the two projects. And then I'm going to talk about the people that are currently involved. Because when it comes to startup technology, the team of people that you put together for those companies is extremely important. Because like I said, oftentimes this technology fails. And when you fall down, the investors hope that they've put together a team that can maybe come up with some other concepts, and look for other ways to make the thing still be somewhat successful. So I'll just shoot up the pictures of each team at the end of each presentation. So number one, it's a company called Ascribe Biosciences. We've put together kind of a pretty funky logo, I think, for it, which we call Revolutionary Crop Protection. And then here's the tagline, which I'll explain in a second. Harnessing metabolites from the soil microbiome for crop protection. So let me explain a little bit about what the technology is behind all those fancy words. And to do that, what I want to do is I actually want to show you some of the data for this particular concept, because I think it's pretty indicative of how this technology works, and I can show you. So ascaricide 18 is a lead compound, and what the heck is that, right? So ascaricides are small molecule compounds that were discovered several years ago now in Frank Schroeder's lab here at BTI in a collaboration that he had with a person named Paul Sternberg, who's out at Caltech. They were studying how nematodes, so nematodes are these small soil-dwelling worms. They're ubiquitous, really, in the soil. So they're all around us. And they're a very well-developed model system for doing genetics and also for doing metabolomics, is what we call looking at what these compounds are. And they turned out to be compounds that the worms produce to communicate with each other. Communicate is one of the things they do. It also has to do with sex determination in the animals. And it's a lot of signaling molecules. So think of them a little bit as like you hear about pheromones in insects. These are sort of the same communication molecules between nematodes. And one of the things that Dan Klessig, who's a plant pathologist here at BTI, and Frank talked about is, these compounds have been in the soil with plants for a long period of time. Is there anything that they do, in the plant world? So they looked into that. And one of the things they discovered was when you expose plants to these compounds at certain concentrations, turns out to be very low concentrations, which is what the worms use to talk to each other with, it turned out that you see the immunity in the plant. Certain genes that are involved in plant immunity are upregulated. So probably the concept here is these plants are sensing these compounds, and it tells them that there's something up. Something's going on in the soil, and we maybe need to kind of put up our defenses. And so that's what we've been seeing when we use these when we expose plants to these things. And so we've done it with a number of different collaborating labs on a number of different plant and pathogen systems. So those are the bad guys. What the company is doing is pursuing, well, I'll get into this last bullet point in just a second. Let me explain what the pictures are here on the left. So what we've done here is we've taken this Asker 18, this worm-derived small molecule, and we've coated soybean seeds at a very low concentration with the chemicals. And then we dry the seeds, we wash them, get away any excess stuff that would be there. And then we wait 10 days, and then we plant those seeds in the ground. And then these are soybean, I should have said, these are soybean seeds. We plant them in the ground, and then we wait for them to germinate. And then 2 days after they've germinated, we expose them to a fungal pathogen, which you can see here killing this particular plant on the left. And then at 4 days, 6 days, and I think 8 days post-infection, we then take photographs of the plants to see how they're responding or not responding. And on the left, these are untreated or mock-treated seeds where they haven't been exposed to Asker 18. And over here on the right are ones that have been. And in this particular case, R3 is a very virulent form of this fungal pathogen. You can think a little bit about this one is similar to like the late blight or some of these tomato pathogens, that cause the Irish potato famine. This is somewhat related. It's a similar type of plant pathogen. And you can see here in this particular picture, and this was done with lots of plants actually, a number of different pots. You can see a pretty dramatic difference between the treated plants over here and the mock treated plants over here. If you're very observant, you can see it's not completely protected from this particular pathogen, but there's clearly a pretty drastic difference between the ones that are treated and the ones that are not. And a lot of time with technology like this, this is a starting point. We got a good feel with especially all these other ones where we saw similar results, that this could be turned into a real product that could be used to protect agricultural crops. But it takes a lot of work. It's still moving it across that bridge, It's like we're still on this side, pretty close to this side of it. And so this was actually done by one of our collaborators who's at the University of Kentucky. So this is good. This is work that was done even outside of BTI. It's always good to see sort of unbiased, not that we're biased here at BTI, but to see other people able to repeat our experiments in their hands. And so, I didn't mention, I want to finish by saying, the other thing the company will be pursuing as well as this lead compound is looking for other compounds in the soil microbiome that could be used to treat plants. So we've given it this name, we call it Precision Soil Metabolomics, PSM. This is really kind of repeating the process that Frank used to discover these compounds, but doing it in a high throughput way where we look for many other compounds at the same time that could be involved in this. And what's good about that is with a startup company, they're often kind of like a one-trick pony. And if the drug target that they have doesn't work, the next day you read about it in the newspaper and everybody's gone home and the company's done. So it just kind of gives you a little extra layer of protection to sort of show that you're going after additional technology that might be valuable as well. Okay, so does everybody understand the concept? Okay, do you guys? So now I just want to give a status update on where we are with the company. Again, I'm going through both of these fairly quickly, but I think it gives you a pretty good idea of what we're working on. It was incorporated as a limited liability company here in New York State in November of last year. We have a scientific and management team that we've partially assembled, and I'll show you who they are in a second. We actually, at this point, have an exclusive option that we're done negotiating and are in the process of executing, actually, with this company. So the Ascribe will have an exclusive option to BTI technology that's involved with here, and then there's certain milestones that they have to meet, and we hope that'll eventually evolve into an exclusive licensing agreement, but we start off as sort of an option, as like baby steps to make sure we protect everybody involved. There's something called SBIR grants. So this is Small Business Innovation Research Grants. These are grants that are given by the same funding agencies that everybody here at BTI hears about, NSF, NIH, DOE, but they're actually targeted at commercial companies that are in the process of trying to develop very new, novel technology. And if you put together a company, you are eligible to apply for this kind of technology. It does require eventually a 50% company person before you can get the funding from these. So you often work with sort of an incubator at the beginning, and then you can also subcontract money back into the lab here to do some of the kind of preliminary experiments for you. We've put together a pitch for potential investors, and we took that for the first time on the road. We went to the North Carolina Agricultural Biotechnology Council meeting in April of this year, where they were hearing from 12 different new startups pitches about new companies. So we talked to them. It's an 8-minute period of time that you have, pretty intense, and then a short period of questions from investors and also from other commercial scientists and others that are there. So it was a pretty interesting opportunity, and it went very well. I think I may have a picture of us doing that, I'm not sure. We have an internal program for funding projects that we call the Triad program here at BTI. And we currently have a Triad-funded field trial for this particular product in wheat using a different fungal pathogen in collaboration with a USDA lab at Washington State University out in Pullman. And this has actually begun. Merle's gone out, treated seeds. They've put them in the ground, and we're in the process of getting results, which actually look quite good for an initial thing. Because, in the previous slide that I showed you, these kind of experiments here, this is really, it may look like dirty soil, but in a sense, this is sterile laboratory kind of conditions. It's not out in a field where you're going to experience lots of different organisms, and soil that's here is not the same as soil that's in Washington. There's all kinds of pitfalls that you can sort of run into when you start testing things in a real world environment that don't seem to behave like they do in the lab. So even to see preliminary data that's positive is good for that particular project. Once we hopefully get some SBIR money, we plan to probably partner with the McGovern Center, which is the incubator here on campus, to set up our first home for Ascribe. And again, like I said, we'll probably be doing work back and forth between the labs here and the company, which would be located down the street. Okay, so that's the status update for Ascribe. And then these are the two pictures I wanted to show you here. I thought I had these here. This is Merle down in RTP making his pitch to the investors. Kind of a cool picture. He's a postdoc in Dan's lab. I'll introduce him a little bit more in the last slide here where I talk about the team. But this is certainly a new environment for Merle to be presenting in. It's not really a scientific environment. It's pseudoscience business environment. So I think it's a really cool opportunity for him. These are pictures of the ascaricide-treated wheat seeds that are ready to go into the field out in Washington. So I just thought I'd show you a couple of fun pictures that kind of demonstrate that we're really actually doing stuff for this one. And the other one too. Okay, who's the team? The current team is made up of really 4 people here associated with BTI. The two major scientists, well, Murley's a major scientist too, but the two project leaders involved here are Frank Schroeder, who I told you about, who's our metabolomics expert, our chemist here at BTI, and then Dan Klessig, who is an expert in plant pathology, is on the team as a co-founder. Jay Farmer is a local entrepreneur that has a lot of experience with several different startup companies in and around the area. Also a real expert on patent strategy. He's worked for a lot of years on patent strategy for a number of different firms. So he's quite good. And then Murley Manahar, who is a postdoc in actually Dan and Frank's lab now together, is also a co-founder. He was a postdoc, like I said, here in the lab, and he's doing most of this work now. And he would transition to at least 50% company position. One of the things with these startups is you sort of need somebody who is kind of willing to migrate with the company to its beginnings. So you'll see in both of these teams, if you don't have this component, it's too early to talk about spinning anything off. So that's just a very important part of the project. Oh, I forgot to mention here. Our first scientific advisor that we've recruited is a woman named Jo Handelsman, who's a soil microbiome expert at the University of Wisconsin. Some folks may have seen her a few months ago when she was here at BTI. She was talking about her science, but she also started her presentation with her work as the Associate Director for Science in the White House under the Obama administration. She worked in the White House as a scientific advisor to the president, which is a pretty interesting position, but it's a very good contact for the company to have. And she really is, they're putting together libraries of soil samples in Wisconsin that can be used to screen for lots of different compounds that are yet to be discovered, and there's lots. So that's where we are with the Scribe. Hopefully that gives you a good idea of where we are. The next one I'm going to talk about is a new company, again, that's called Effector EX. Here's a picture of something that's related to the concept. And this is going to take a little bit of time to explain what the concept is here for this particular startup. This is a neat picture. These are Pseudomonas bacteria. It's a type of plant pathogenic bacteria. And they're here. This is probably a tomato leaf, but they're on the leaf of the tomato. And you can see them trying to sneak into the stoma, the stomata, the openings where the plants are breathing. This is a very serious plant pathogen for tomatoes and some other plants as well. So now I'm going to explain what it is, and this may sound like you don't understand anything, but don't worry. The next slides hopefully are going to straighten it all out. What we're doing is trying to improve recombinant protein expression by leveraging natural effector proteins. I know that probably doesn't make too much sense, but I just want to remind everybody, we use a number of different organisms to make recombinant proteins. I use one of the easiest examples that people are usually heard of are influenza vaccines. So that's a protein. The vaccine antigen is a protein, and it's made in chicken eggs. In most cases, there's a few other flu vaccines that are out there now, but that's one way to where we make recombinant proteins in other organisms. You hear a lot about proteins that are made in yeast, fermented in yeast or bacteria. We also make proteins like vaccines in human cell lines. We also make them in insect cell lines, like the Cervarix vaccine that I described to you in the beginning of the talk. But another way to make recombinant proteins is in plants. Again, I mentioned the Dow AgroScience project that we had making vaccines in plants. So that's an example of making recombinant proteins in plants. And I'll explain how we do that. And then effector proteins are proteins that come from the bacteria in this particular case. And one of the things that we use to try to describe what goes on between plants and these pathogens is a molecular arms race, right? The plants develop mechanisms to defend themselves against these pathogens, and the pathogens in response evolve ways to circumvent those defense mechanisms. And the one that I'm going to tell you about here, we call AVR-PTOB, which we patented a few years ago from the Martin lab, is one of these effectors. And I'll kind of just briefly describe it, and then you'll understand when I show you a few other slides kind of how this particular one works. One of the things that plants do to defend themselves when they're being attacked by a pathogen is they kill the cells in the area where the pathogens are. It's called programmed cell death. So the plant senses that they're under attack, and in that area where they're being attacked, they kill the cells because these pathogens have to have live cells to really effectively infect the plant. And so they do that, and the bacteria have evolved this protein called AVR-PTOB, which is a plant programmed cell death suppressor. So when it infects a plant, and the plant tries to turn on this programmed cell death response, the bacteria turns on this anti-programmed cell death response to try to suppress that activity and kind of give them a way to get in. I think about it as like, the garage door is closing and you're trying to crawl underneath, and they're sticking a stick in there to hold it open, and so that these guys can run in. So that's kind of a funny analogy, but that's kind of a neat way, I think, to think about it. And then I'm going to talk to you in these other slides about how you use this to improve recovery of proteins in plants. But let me just help you. I want to just a few pictures here on where the system comes from. Greg studies this disease called Pseudomonas syringae, which causes bacterial speck in tomatoes along with other plants. This is a picture of what the plant disease looks like. It causes these specks on the fruit, on the flowers. You see it here on the stems, over here on the leaves. And this is an economically important disease in a sense that it's not good for people that are trying to grow tomatoes when this is around. But it's a very good model system for understanding how these bacterial pathogen, these bacteria, and the plants evolve, like what I told you about. These are where these discoveries that Greg has made come from. So that's the system where we've made sort of these discoveries. But to fully understand that, let me just back up for a second and explain how we produce proteins in plants, because I think it's still a little tough to understand how you actually do that. So I'll give you sort of the steps that are involved. This is a picture of crown gall. Most people have gone around and seen these type of things on trees, right? These are actually caused by a bacteria called agrobacteria that infects trees out in the wild. This is I'm not sure exactly how these galls work. It's some sort of a defense mechanism for the bacteria. But it's just sort of interesting. I know if you go across the narrow bridge over into Forest Home, there's a tree there that's just covered with these galls, and those are probably agro-caused galls. There are some other ones that are caused by insects and stuff, but a lot of times this is what causes. So what about agrobacteria? Who cares, right? Well, I'll tell you what we use agrobacteria for. We use it to move proteins into plants. Okay, so like I said, agro is this bacteria that infects plants. So in a sense, we've hijacked that system to bring the genes and the proteins that we're interested in into the plant system. And the way it works is, here's a picture of agrobacteria doing its natural thing. It's infecting a plant, right? And it would normally shoot in all of its DNA, all of its genetic material into the plant that then gets replicated, and then new bacteria or viruses, whatever, are formed, and then it kind of goes out. That's part of their life cycle. But what we've done here is we've taken a thing called tDNA. So this is a gene of interest. Let's say this is a gene that codes for vaccine A, right? Whatever. You can engineer that into a plasmid, which is just a piece of circular DNA that allows you to put DNA into a bacteria. We use this all the time in the lab. And then again, the bacteria does its natural thing. It infects a plant cell, it shoots the DNA in there, and then that can be replicated inside the nucleus of the plant cell, and then you can somehow, a couple of different ways, recover those proteins from the plant. You could grind it up. You could do it in some ways where you're working with plant cells in culture, where it secretes it into the media. There's a bunch of different ways to do it. One of the common ways to do it is this picture over here. It may look somewhat crude, but what we're doing is we've got a syringe with a buffer that's got our agrobacteria, that's got our gene of interest in it, and we're actually just physically squirting it into the plant. This is what we call transient expression. So what we're doing is we're not making a transgenic where the whole plant, all the cells can produce that particular gene of interest. We're just kind of hijacking one part of the plant to put in our constructs so that we can generate plants. But what happens is the plant will take up this construct, and then these leaves where we've done this over time will generate that protein, and then you can take those leaves and purify the protein that you're making from them. And so, we say the plant as a protein production factory. I give people the example, since many people have heard about the Ebola outbreak that took place a few years ago. If you heard about that, there was a miracle cure that they were working on that was called ZMapp, which was an antibody treatment actually for Ebola, which is in clinical trials right now. And that particular treatment is made in plants. You need a lot of this antibody. I guess the way it works is you take antibodies from Ebola survivors that had a good immune response to the virus, and then you grow those antibodies up in a system, in this case plants, and then you give that to patients that are to have a positive reaction to the virus, the treatment anyway. So that's one example, but there's a number of other examples out there. Of course, the plant-made vaccine example, which I gave you at the beginning, is another one, and that particular one was done in transgenic solution of plant cells, but a lot more work is done in this transient expression level. But again, what you run into here, which I'll show you in a few slides, is the plant sees this thing you're trying to squirt into it as a threat, right? And so it turns on some of these natural defense mechanisms. And so that can be a problem for people that are trying to make proteins in plants. And then I'm just going to quickly go through this slide. I've told you AVR-PTOB suppresses programmed cell death in plants. This is just an example of what that looks like. So what we've done here is we've got a control leaf on this side where we haven't done anything to it. We've got a bacterial, a fungal, and an animal inducers of programmed cell death. This gets a little bit complicated, but I think you can follow me. In here, we call it an empty vector. But basically what we've done is we've shot our gene of interest into the plant, and we see how it responds. Normally what you get is this programmed cell death response. This is called hypersensitive response. If you've looked at leaves out in the wild, a lot of times you pick up a leaf and it's got, we call this chlorotic lesion, where it's got like a spot that looks sort of yellow or whatever. It's a very common thing in a lot of different types of plants. So you see that sort of natural defense mechanism working here. But here we've put in examples of these inducers of programmed cell death. And when we put in AVR-PTOB at the same time, we see that cell death suppression go away. So all of a sudden these leaves that are there, when they've got AVR-PTOB, you start to see that you don't see that chlorotic lesion. You don't see that programmed cell death happen. Now, how do we use that in sort of a practical biotech way? That's just showing naturally kind of how the system works. One of the things that happens, here is a slide where we are actually expressing a vaccine capsid protein. So this is done in collaboration with Hugh Mason, who's out at Arizona State University, who works on plant-made vaccines. And what they've done here is you are trying to express, in this particular case, this is, if I remember right, this is a norovirus vaccine that they're trying to make in plants. And when you put it into the plant and try to get it expressed, you can see what happens. It's kind of a disaster, right? Programmed cell death response is really strong, and you can't really make any, you certainly couldn't harvest this leaf to make much protein. What we've done here is we've put in a couple of different amounts of AVR-PTOB to see if we can suppress that programmed cell death response. And it looks pretty good. These plants, they look very healthy, but what's happening here is these plants are taking up this vaccine construct, and they're going to generate protein. And so I'm not showing it on a slide here today, but if you look at a graph of once you've purified protein from these leaves, you see a much higher rate level of protein expression. in these particular types of ones where we've co-expressed AVR-PTOB. So hopefully that explains how this works. The next step from sort of a commercial standpoint is, there's a number of companies that are working on recombinant protein expression in plants. They're the target for these particular applications. So it's very different than like, the seed world of soybean treatment. Here we're talking a very different concept where you're going out to companies that are trying to do this biotech process. And we want to use our technology to improve that particular process. So that's the way this particular company would be set up. And again, this is a very new company, very virtual in a sense, where we've gotten this preliminary data where we know in some systems we're seeing some response. The question now is what do we put together as a business plan and how do we market that technology out? And so to give you the status update on this particular one, we've got this preliminary data showing the improvement. Actually, we also did it with a hepatitis B vaccine where they run into issues with programmed cell death, limiting their expression. And that's all being done in collaboration with Hugh Mason, who used to be a project leader here at BTI when we were working on that Dow AgroScience project, for example. The company's not yet incorporated. It's very new. What you need to be is incorporated once you want to collect any money from a program like SBIR. So you can really sort of conceptually work along until you get to that point. But then you eventually have to take the leap into a company. We've engaged a consultant to help us draft SBIR applications. These are very specific sort of grant applications. We normally are writing grants for NIH and NSF, and we don't talk about commercial anything. We may talk a little bit about broader impacts or things like that, but we don't talk about markets and money. And so that's what you have to talk about in an SBIR grant like this. How would you make this science into something commercially viable? So we need to get people outside who have that kind of help, that kind of expertise that can help us. Fabio Rinaldi is a postdoc in Greg's lab. We had him put together a poster and a talk actually where he went to this protein engineering summit in Boston in early May to present the technology, kind of get a feel from companies and people that are doing this kind of work, what sort of value they might see in this. There's actually a part of the meeting is called difficult to express proteins. So it's kind of like it's a whole field of coming up with different ways to try to improve protein expression. And this is a fairly novel, I think, approach to maybe solving some of those problems. And this came across very well. We actually have several companies that have contacted us already about wanting to test the technology. And we're like, whoa, wait a minute, we're not quite ready. But anyway, so that's going well. We're in the process of recruiting a scientific advisory board and really figuring out who would help us manage the business side of this particular project. And again, we talked about the one trick pony thing. We're also looking at maybe developing a platform for discovering new effectors, because there's lots of different proteins that are made by these organisms that do different sorts of things that could potentially be harnessed in a biotech way to improve protein production. Some plants, PCD is an issue. Sometimes it's not, actually. But there's other things, too, that you run into that could be solved by other effectors. And again, that would give you sort of a broader scope for the company so that you have several areas you're working on at the same time. So that's where we are. Okay, so what does the team look like? Oh, this is just a picture of the cover shot from Fabio's presentation that he gave down in Boston. These are actually just a number of plants that are actually, this is sort of what, not very fancy looking, but this is what a protein factory looks like when you're doing transient expression in plants. It looks like a room filled with plants, basically. Most places, when they get into commercial levels, there are robotics and everything involved, to make it at a very high throughput scale, but that's really what protein production in plants looks like. It looks like a bunch of plants growing in a greenhouse. So here's the people that are involved. Greg Martin is one of the founders of this. He'll be one of the founders of this company. He's been at BTI for many years. He's an expert in plant disease resistance. He discovered AVR-PTOB along with a number of other effectors. Like I said, this is one that we've patented specifically for this particular use in plants. Fabio Rinaldi is a postdoc in Greg's lab. Again, like I said earlier with Murley and a scribe, you really need sort of a person in the company who would be a point person for the company once you want to make that sort of move to the next to the next level. So Fabio has a lot of experience in recombinant protein production, so he's really a perfect fit for this. He understands plants, but he also understands making proteins in a bunch of different ways. And then this is a picture of Hugh Mason over here, who I worked with quite a lot when he was back here at BTI, but again, he's an expert in plant-produced vaccines. He's at the Vaccine Institute, the Biodesign Institute at Arizona State, where they do a lot of this kind of work. He actually published the first peer-reviewed paper on plant-made vaccines back in '92. So he's sort of been in this field for as long as it's existed, in a sense. So he's really a great guy to be working with. I want to hear, it's important for me to acknowledge the folks at BTI and where the money came for these, the funding has come for these projects. This is Eric Richards. He was the past VP for research here at BTI, who oversaw the technology transfer program, was my supervisor. In the middle here is Kelly Montz, who's our technology transfer specialist. She works with a lot of the day-to-day workings of the tech transfer program here at BTI. Over here, this is a picture of David Stern, the president of BTI. This is actually him out in Butte, Montana, where he went out to give a scholarship to this girl here from one of the funding programs here at BTI. But David has always been a real proponent, along with the board here at BTI, of tech transfer and having it be a big part of our program. Greg Martin's a current faculty rep, but everybody's involved with tech transfer at BTI. All the administrative staff, the support staff. Kelly Aylesworth, who used to work with me on a lot of tech transfer related stuff. You know, it's really a whole institute program, in a sense. None of it could work without all these different pieces coming together. And then interestingly enough, I think here's a long list of things that have helped us get to where we are. The Triad Foundation has helped with supporting several of these projects, especially the Ascribe project. They've had 2 Triad funded grants now, which have helped bridge us to USDA funding in Dan's lab. They helped us get to a collaborative research funded agreement with BASF, which we had a few years ago. The Institute has spent money on these projects as well, certainly spent money on me working on these projects. And then NSF and NIH there was grant funding from NSF and NIH on both of these particular projects as well. All right, well, thanks for your time.